welcome back. This is chapter 13. You don't actually need to finish this week's stuff up until after spring break, but I'm posting it the week before, and I want it, technically it would be due the Monday of spring break, but that seems mean. So I'll make it due the Monday after spring break, so you have a little bit of time, but that means you have a little bit of time to really think about it too. I also want you to go back in time and look at your bio notes for transcription. Transcription is the process of making mRNA from DNA. I think you did some of this. I think you did bacterial transcription. So I'm going to talk a little bit more about the history of how this was understood than you covered in bio. And then I'm also going to talk about a little bit more about how bacteria do this process and how eukaryotic organisms do this process, and some variations. Today, there's going to be 2 videos for this lecture. We're going to talk about the genetic code and the history of understanding the genetic code in this lecture topic, so this video. I'm going to talk about particularly things like what does it mean that the code is degenerate and unambiguous. I also think as you're thinking through this, remember that when we use these nucleotides to send information, they are paired using hydrogen bonds in a complementary but anti-parallel structure. I want you to go back and review what's the difference between RNA and DNA, because I don't cover it again. But RNA is structured a little bit differently as a molecule. It also uses the nitrogenous base uracil instead of thymine, which is what is used in DNA. So a little bit of a review of that, especially if you're watching this at the end of spring break and it's been a week or 2 since we've talked about DNA. Go back and quick review those things, because I want to talk about how we actually use those 2 DNA and RNA together in order to make a piece of information able to leave the nucleus and head off to the ribosome. Chapter 14 and 15 is about the specifics of translation, doing those things in the ribosome that turn things into protein, and then what happens next. In the second video, we'll talk about the specific sequences for transcription. So, how is your genetic code stored? And I use this word encoded all the time. It just means that it's kept in code. You don't need the z there either. How is genetic information encoded? And where do we keep it? It's encoded in the DNA. And so you keep that as a eukaryotic organism in your nucleus. But if you are a bacteria, you would keep it in your cytosol, just floating around in your nucleoid. How do we actually get DNA, which is just storing information for the entire organism, into RNA form so that it can be understood by the ribosome? That's transcription. We're going to go ahead in topic 14, which will be next time, not this week, but after spring break. We'll look at translation, which is the process of taking RNA and converting it into a polypeptide. The genetic code uses a series of rules that are uniform across almost all living things. When I take a piece of DNA and convert it into RNA inside my body, each nucleotide of RNA is kind of a letter. Basically a letter in a word. Every word that we make reading the DNA into RNA is a series of 3 letters. It's called a triplet codon, a 3-letter code. That series of 3 letters is unambiguous, meaning it means the same thing all the time across almost every living thing. I'm going to show you some weirdos that vary from that, but basically all living things use the same basic template of information. It is also degenerate, which makes it sound like it's breaking down, but that is not what this word means. This means that there is a little bit of redundancy in it. And I don't know the historical reason for not just saying there's redundancy, but degenerate means redundancy. If these words that we are writing about the story of what kind of protein I need you to make is a sentence, the sentence has a start and a stop. It has a beginning, the start codon. is a codon, and it's actually coded for by the three letters AUG. That's where the molecule, the ribosome knows to read the molecule as start. The sentence that we write also has a stop codon. There are three versions of the stop codon that tell the ribosome that it is done with this particular polypeptide sentence. There is no internal punctuation, so there's no commas, there's no semicolons. Everything is one giant sentence made up of words that are 3 letters long. Each set of 3 letters is made out of 3 nucleotides of RNA. The code itself is collinear. Collinear means that the order of the nucleotides actually corresponds to a corresponding order of amino acids. There's no resorting after the fact. We don't rearrange them and create new sentences. Once that is taken into the mRNA and it goes to the ribosome, that is the final sentence, and we read it in order, and it's mostly universal. I think in Bio 1 you probably talked about it as being universal. I'm adding nearly here because I got a few weirdos for you that read it differently. And you know, we find a couple weirdos. That's interesting. As time goes on, we find a few more that teaches us more about how the code actually works across living things. So I want to talk a little bit about how we understand the triplet nature of the code. Francois Jacob. Francois Jacob and Jacques Monod postulated the existence of mRNA when DNA was still trying to be understood. We understood DNA from the mid-1950s, like 1952 or 1953. We understood the structure of it. But the actual looking at it and understanding how it worked, we knew how it copied, but not how it conveyed information. These guys thought that it must be a series of lettered codes inside of it that talked to another molecule that then sent it to the ribosome. And they actually thought that it was built that way because the DNA itself is in the nucleus surrounded by a phospholipid bilayer. And that phospholipid bilayer is semi-permeable. Things can't get in and out very easily. When I'm inside a nucleus, that DNA is kind of trapped in there. It's stuck. So they hypothesized that there must be a second molecule, one that could leave. We now know this is mRNA. mRNA could leave to go to the ribosome because there had to be a mechanism of transfer of information. Now they also had the same problem that people early on were worried about in understanding how DNA worked. We're still faced with this problem that there are 4 nucleotides in DNA, A, C, G, and T. In RNA, there's A, C, G, and U, but we have a code made up of 4 different variations that needs to code for 20 amino acids. Now, way back with Hershey and Chase and everyone earlier, the discussion had been, how could this possibly be an information molecule if it only had 4 nucleotides? Now the conversation was, how does this translate into something more complicated? Sidney Brenner suggested in the late 1960s that the code has to be in 3s. Because if you had a code where there were just 2 nucleotides, you could have A and G, and then A and T, and then A and A. This actually, if you play out all of these possibilities, they only end up coding for 18 or 16 amino acids possible. So this is too few. That's too few. A code of 4 could code for, if you had a code of 4, it would be A, G, C, T, A, C, C, T, and so on, all possible combinations. There's actually 256 combinations there. That's too many. If you calculate the sets of 3 and you want them to code for 20 different things, a code of 3 is just right. It has 64 possible codes for amino acid. That gives a little bit of redundancy, but covers all the possible amino acids without too many extra codes. It's a Goldilocks situation. So this is the three bears. This is a strange, these are three different species of bears, but these three bears and Goldilocks, when we talk about in the sciences, when we talk about something that can't be too much and it can't be too little, it has to be in a zone that is just right. We actually describe it as a Goldilocks phenomenon or a Goldilocks situation. I don't want to say situation, but it is. This is a Goldilocks solution. But we still only have 20 codes. We have 20 amino acids, but now we've got 64 possible combinations of nucleotides that make up those amino acids. Crick, and yes, that Crick from Watson and Crick, and a whole bunch of other people, and in your book, it lists all of their names, and then some of the contributors. I've tried to grab some of them for this lecture. Crick and other people actually showed that the code was in triplets definitively. And so I want to walk you through some of that research just so you can see why and defend, frankly, why it's a triplet code. Crick actually had started out with his research using frame shift mutations. Frame shift mutations are when we have a code. I'm going to pull G A G G A G A G G A here. G G A. A frame shift mutation assumes that the reading frame is made up of sets of three. G A 1 2 3 1 2 3 1 2 3 G. A set of three. If I add something to it, I stick in a C here, which is what's happening over here in this picture, but I want you to see it in action. If I insert a C there, I change my code to G-A-G. That doesn't change. C, G-A-G, G-A-G. I change my code by adding this new nucleotide. And because I have to, as a reader of genetic information, read in triplets, my first word stays the same, but my second word becomes a new word. CGA codes for a different amino acid than GAG. 1, 2, 3. 1, 2, 3. This causes my reading frame to shift. And so now my first amino acid is the same, but this one's new. And also 3 is new. I have now a completely new set of reading codes that I read. I can fix this by having another frame shift. So this is a frame shift, which is a reading frame shift. If I add or remove, let's see, I remove that one accidentally, another mutation. GAG is not affected. CGA is my new one. And then GAG 1 here stays the same because nothing happened to it. 2 is new because it had an addition into the frame shift. 3, because it lost this G, returns back to its original code, and that means with an addition and a deletion in 2 separate places, I could end up with just one amino acid off sequence. A frame shift mutation can be devastating if it occurs with an addition or a deletion once, but if a second frame shift in the opposite direction happens, we can restore the reading frame. When Crick and friends experimented using frame shift mutations, they used it to show they could alter the types of amino acids that a sequence coded for by adding or deleting one of the nucleotides. The other major way that the code was elucidated, understood, was by Nuremberg and Mattei. They synthesized strands of DNA that were homopolymers, And I think this is worth pointing out because we had talked the other day about how frequently the prefix homo and hetero appear. It's been a while. Homo means same. Polymer is a sequence of mers. Or a sequence of monomers, single pieces. The sequence of DNA, the sequence of RNA is a polymer. It's a building block, building lots of monomers of the nucleotides together. Nuremberg and Mattei actually sequenced strands of RNA that were homopolymers, or they were made up of all of the same monomer repeated over and over and over again. And then they synthesized these in a lab into chains of amino acid polymers. I better write out amino acid here. Amino acid polymers. When they did this, they got lysine for AAAAAA. They got lysine, 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 lysine. And so that led them to discover that AAA is the code for lysine. For UUU, they got phenylalanine, 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 phenylalanine. So they understood that UUU codes for phenylalanine. CCC, the same thing. It was proline, 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 proline. GGGGGGG. This one actually doesn't work. If you do this kind of as a side note, what happens is the molecular structure of guanine actually causes this to loop-de-loop on itself, and it won't fit into the ribosome. So this one actually doesn't work if you create a homopolymer. We had to wait until much later. Other ways were discovered in order to figure out what the sequence of GGG was. I do have two things at the bottom here, if this is your thing. The legendary Thai club, where everyone was assigned an amino acid and then sent off to try to figure out what sequence of RNA coded for their amino acid. This was partially tongue-in-cheek, but is a fun read if you want to take a stab at this, about the role of community in science, and also how people were trying to figure out lots of different ways to figure out this code that we couldn't see, but we knew the outcome of. This here is Nuremberg and Mattei's paper, which is incredibly accessible for you as a genetics student. You already know all of the stuff inside of it. You could just sit down and read this, and it's their Nobel Prize winning paper. There's also this idea of a triplet binding assay. I want to introduce this partially because this is where the next lecture goes, and so it gives you a little bit of flavor of how these things connect, but it is also the way we can discover something like what does GGG code for, because the other methods don't work for that. What you do in a triplet binding assay is you take a ribosome. Ribosomes are made of a large and a small subunit. I think you remember this from micro and from bio. The small subunit and the large subunit differ in catalytic regions, but also in size. The mRNA is slotted through between the small and the large subunit. And then it's read in triplets. 1, 2, 3, 1, 2, 3. It would be like G, 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 G. GG. Or U, or AAA. We'll do a bunch of them. There is also the addition of, if you're trying to read in a ribosome, you put the mRNA through it. The ribosome, which is also made up of RNA, it's made up of rRNA and proteins, clamps around the mRNA. And then the triplet code is read by a third kind of RNA called transfer RNA, or tRNA. And we mentioned it briefly as I talked about RNA when we were talking about the structure of molecules that retain energy or information. tRNA molecules enter into the ribosome, and they form a complementary bond, a hydrogen bond, with a triplet code on the mRNA. So if the mRNA's code reads UUU, the corresponding tRNA, I'm going to just make it T-shaped, the corresponding tRNA that matches with an complementary set of RNA nucleotides. On the other side of the tRNA, there is the amino acid that this triplet code on the mRNA codes for. So the tRNA reads the mRNA by binding to the codon on the mRNA with a matching complementary code. We call it the anticodon. So codon and its opposite, the anticodon. When they form a strong set of hydrogen bonds, that actually catalyzes the reaction that moves the amino acid into the growing polypeptide chain in the ribosome. We can use this in a triplet binding assay by radioactively tagging the tRNA itself and the amino acids, and then looking at how, when we put these guys in a centrifuge after allowing them to react with each other, looking at where the radioactivity ended up. I am going to talk about this again as we get into looking at how the ribosome works. I just think it's helpful to hear it now and then hear it again. I actually know that you have heard this before in micro and in biology, so a little bit of a refresher, and then I think it'll be awake in your brain. When I read my mRNA, I can actually read it as a human being by using this genetic coding dictionary. So this one. This genetic code, because the code itself is used the same throughout almost all living things, we can, after doing all this research and finding out what each set of triplets coded for, we can make a map showing what code aligns with what amino acid. So these are the triplets. These 3-letter words right here, Phi, Lu, Il, Met, Val, those are representing the amino acids that these letters code for. The way you read this is, if I have a code that says C, 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 I start reading from the 5' end of my molecule, my mRNA molecule, and I read C. That's my first position. I read my second position, C, here to second, this is second position to C. And then my third position, I find it in this box. That leads me to this amino acid. Now you note that if this amino acid, it didn't even matter what this third code was. This third code is redundant in this case. It is a bit wobbly. The third position here doesn't actually matter what nucleotide is there. No matter what I do, if I have a CC, I get the same amino acid. I also can see some other patterns that maybe you haven't talked about in your other classes. I can see that things that start out with the same 2 letters, or the letter that are close together on this code, in this chart, actually make very similar chemically amino acids. I'll have things that together, AAA versus AAG, AAA, where is it, versus AAG. These both result in charged amino acids. AAU and AAC also results in a charged amino acid. So one that's a little bit negative or a little bit positive, they're structurally similar to each other. I also want to point out this one right here. This is the start codon. The process of translation, when mRNA is being read by the ribosome, will not start until the sequence AUG in order is found. That means that as I stick my 5' end into the ribosome, It could be U, C, C, A, U, G. It will read through this, but the ribosome will not start the translation process until I read A, U, G. I do want to point out too that if I flip this and I gave you a code that looked like this, G, G, C, A, G, U, A, I don't want to make it too long, C, C. If I'm reading this code, I still have to read it from 5 to 3. I just wrote it backwards on the paper. The ribosome is going to skip the beginning, find A, U, G in this sequence, and this is going to be the start. You start from the 5' reading the mRNA transcript, and then you look for AUG. That places this amino acid. That's the beginning of every protein in most living things. There are also 3 stop codons. 1, 2, 3 here. These three stop codons stop translation. They tell the ribosome to disassemble, basically. Every code for a protein on an mRNA transcript starts its coding of amino acids at the sequence AUG, and it ends with one of these 3 stop codons. In between here is the actual code for the gene or the polypeptide that we are making. The stuff at the before and the stuff at the beginning and the end, these are irrelevant. It doesn't matter. Very much like a computer program. You can put notes at the beginning until you tell the computer to start the process. It doesn't do anything. It will continue until it reads end. Then it stops the process. It doesn't matter what you write after the end signal in a computer program. The code is nearly universal. Some bacteria, actually a few eukaryotes, there's a ciliate that your textbook talks about that's like really weird. I didn't include a bunch of stuff about it, but check it out if that's your thing. A few eukaryotes and a lot of our mitochondrial DNA have variations in the code. I think that here you look at human mitochondria here, the codon UGA becomes, it normally is a termination. It actually codes for an amino acid. In the paramecium that they talk about in the textbook, UAG, which would normally be a stop codon, becomes an amino acid. These codes have slightly different meaning. They do not seem to be related to each other. Mitochondria and paramecium do not share a common recent ancestor where they have the same kind of mutation. These seem to be mutations that have carried and independently evolved in these different branches of life. In bacteria, we have a case where sometimes we can find genes that overlap. Not in eukaryotes. In bacteria, that we can have a single mRNA, and it might have start points at various spots. There is more than one polypeptide coded on this gene. The difference between this polypeptide and this polypeptide is just which AUG the bacteria's enzymes choose to start the process at. We also see open reading frames, which is when the AUG positions are not the first AUG, but alternate AUGs are allowed to be start transcription. This does not occur in eukaryotes, only prokaryotes. 2 reasons. This does make everything a lot easier if you have just a little bit of DNA. It means that you have fewer opportunities to make a mistake during DNA replication because your DNA is smaller. And if you only have a few different polypeptides that you're making, this is going to work fine. But flip that. If you make a mistake in your DNA replication process, you can affect multiple genes in the genome instead of just making a mistake and causing one gene to have a problem. And for something like a eukaryote with a lot more genes and a lot more complexity, making a single mistake that affected multiple genes could be completely catastrophic. This is not a functional solution for anything in eukaryotes, but it's okie dokie enough for a prokaryote. So a little bit of practice with me. These words here are all words that I want you to be able to define on the exam. And this is the third exam if you're watching this a little bit early. So the third exam it's in April, I believe. The genetic code is blank. This means that an amino acid may be encoded by more than one codon. Degenerate. Degenerate means redundancy. I wrote out my explanation here in case you're flipping through and saving the pictures. I will not put these in the one I give you to read through while you're watching the lecture, so you have a chance to answer the questions. Many amino acids are represented by more than one codon. The wobble hypothesis predicts that codons coding for the same amino acid may differ where? I'm going to go on. At the third position, the first position, the second position seem to matter. For most amino acids, the third position is a little bit wobbly, and we'll look at how that works with tRNA as we talk about translation later. With that, I am on to part 2, so I will end the video here and upload it. And the next video is a little bit longer, I am sure. It's talking about the actual process of transcription instead of the basics. Get video 1 down in your head and what all those words mean. Go back and review what you learned in micro and bio 1 about transcription, and then come back and watch video 2. And I'll see you in the next video. Probably before I see you in real life again. All right. Bye-bye.